Hello, everyone. How are you? Thank you for joining again for the second lecture of the undergraduate immunology course, BICD140. Okay, today we will be covering, starting to cover innate immune responses. So we will go through generalities of innate immune responses, which cells mediate the innate immune responses, how does the innate immune system work and we have talked about some of these generalities and the cells of the immune system as well in the previous lecture, and we have also mentioned that the innate immune response can be subdivided into different phases. One that is preventing the infection, recognizing the microbe, then alarming other cells and tissues of the presence of the microbe, and then ultimately destroying the microbe or the cells that are infected with the microbe. And so here is a nice a graph indicating how invertebrates are the only, invertebrates and vertebrates have an innate immune system, and while the adaptive immune system is only present here in the vertebrates in blue. And it's interesting because there is these jawless fish, which actually are vertebrates, right? And they have evolved an adaptive immune system that is a little bit different from the adaptive immune system present in other vertebrates, including mammals, but it has been convergently evolving adaptive immunity, which talks about the needs for the evolution of adaptive immunity to defend against certain pathogens that were present or were infecting vertebrates, animals. And so we will learn now on which are the cells that mediate the innate immune response. And I'm talking here about the professional innate cells. And so we learned last lectures that all the cells of the immune system derive from hematopoietic stem cells in the bone marrow, and that we learned how most of the cells of the innate immune system derives, these professional innate immune cells derives from the common myeloid progenitor, and including neutrophils, eosinophils, basophils, monocytes, and dendritic cells. However, I want to make you here some comments because there is a more recent literature that shows that some subsets of dendritic cells can also derive from these common lymphoid progenitors, okay? So that is something that I think that you are ready to remember, that the dendritic cells can derive from the common myeloid progenitors, but can also derive from these common lymphoid progenitors. And there is more ongoing research on understanding further these developmental stages of dendritic cells and developmental origin of dendritic cells. We also learn how natural killer cells, which also are professional innate immune cells, derive from common lymphoid progenitors. And so here are the major or professional innate immune cells populations. So we have the natural killer cells, as the name indicates, are cells that have specialization for killing infected cells, particularly virally infected cells. They also play an important role in protecting us against tumors. And so, and we'll learn more about how they work, but they have granules here that induce lysis of virally infected cells or tumor cells. The neutrophils are, their role is phagocytosis and killing of microorganisms. The eosinophils and basophils, that we will learn more about them in later lectures, particularly in the lectures on hypersensitivity and allergy. But basically they will release the contents of their granules to defend us against parasites. And then we have the dendritic cells that not only they will secrete soluble mediators that will limit the spread of any pathogen, but they also play an important role, and we will learn much more about them because they are important for activating T cells and that is necessary for initiating the adaptive immune responses. So they are often known as the bridge between innate and adaptive immunity. And then we will also learn more later about mast cells because they also play an important role in the defense against parasites, and they also play a role in hypersensitivity or allergy, and also is because of the release of granules that they where they contain histamines and other active agents that, for example, induce the muscle contraction that will help expulsing parasites from the intestine, for example. These big warm helminths that macrophages are not able to uptake because they are too big, they will be fighted off by contraction of the muscles and peristaltic movements in the intestine to expulse them from our body. And mast cells are very important for that via the release of histamine, and we will learn a lot more about that. Then we have the monocytes, which we learned they are circulating precursors for cell macrophages, but also can differentiate into dendritic cells. And then we have the macrophages that are phagocytic cells and that have contained in their lysosomes factors that help killing microorganisms. And with less efficiency than the dendritic cells, the macrophages have also been shown to activate T cells, and only after the dendritic cells have done it can help initiate the adaptive immune responses. So the dendritic cells are the most professional and powerful antigen presenting cells, are an essential to initiate adaptive immune responses, but the macrophages are also adaptive antigen presenting cells and can also help initiate adaptive immune responses, particularly T cell responses. Okay, so this is here again the innate cells that we will focus most on today. As I said, the eosinophils, basophils, and mast cells will be covered in later lectures, but in this lecture today, and mostly in the next lecture, we will focus on these cells. And so, as we said, the neutrophils are phagocytic cells. They contain reactive oxygens and nitrogen species that will oxidize factors in the pathogens, inducing their degradation. And they also release antimicrobial peptides that can form pores in the membrane of the pathogens, inducing their lysis. Macrophages also are very important for phagocytosis. They are also important for release many inflammatory mediators. We talk about how they can participate in antigen presentation. So they are antigen presenting cells to activate T cells. They also have reactive oxygen and nitrogen species in their lysosomes where they can destroy the pathogens. And again, they will produce cytokines, which are these inflammatory mediators as well. And we will learn how they can be empowered by the complement, and we will learn more about that in the next few slides. And later in the course, we will learn how they can be empowered as well by T cells. Then we have the dendritic cells, which again, they are known as bridge between innate and adaptive immunity, or they are known as professional, most powerful antigen presenting cells. And this is because they express constitutively, and they can even enhance the co-stimulatory molecules that basically they give the T cells very key signals that they need to become activated. And we will learn about these signals in details and which are the receptors and ligands and cytokines that participate in that signaling. They also have reactive oxygen species, and they can produce inflammatory mediators like interferons and pro-inflammatory cytokines, and we will learn more about this. We also have natural killer cells, as we mentioned, these lyse viral infected cells or tumor cells. They can produce interferon, although a different interferon that is the ones that the dendritic cells produce, that these are interferon gamma, and we will learn more about that. And this interferon gamma can also empower the macrophages. So the natural killers can empower macrophages by releasing interferon gamma, and we will go into that in the next lecture or the following one. Okay, so a nickname for this neutrophil is walking bomb. So the neutrophils will act very rapidly. They are in the blood, but when there is inflammation, they will quickly go to the inflamed tissue, and they will uptake microbes. And also, more recently, it has been shown that neutrophils are able to do something that is called netosis. And it's basically, they will release their nuclear content, and so the DNA that is inside the nucleus, and they will form a net to limit the spread of extracellular bacteria. So we don't have time to cover the details of that, but you can, if you are interested, you can search more about that. It's called netosis, from net, N-E-T, net, netosis, and it's because it seems by microscopy you can see how it seems like they form a net with the DNA that they release to capture extracellular bacteria. Then a nickname for macrophages is garbage collector. And the reason is because macrophages are specialized in phagocytosis, in uptaking dead cells, microorganisms, and we can see how these can be exacerbated, for example, by complement proteins via opsonization. In the next few slides, we will learn that. The dendritic cells nickname is detectives because they are distributed in different tissues throughout our body, not only in lymphoid tissues, but in many different tissues throughout other bodies, in the mucosal mucosa, for example, that is underneath the epithelium in our bodies. And the reason for that is because there they are surveying for the presence of pathogens, so they can, when they see a pathogen, they can release factors that limit pathogen spread and then uptake this pathogen for presentation to the T cells. And so that's why, because they are surveying everywhere in our body for the presence of pathogens, they are called detective. And then we have the natural killer cells, which are also nickname is border patrol, because they would, if they find a viral infected cells, they will go and kill these virally infected cells. So, okay, so that's about the cells, these professional innate immune cells. So do you guys have any question about that? Okay, so this is just an overview, because there will be many more details that we will learn about how these cells operate. But some generalities that we covered last lecture too are on how the innate immune system work are via these three phases, right? Preventing, recognizing, alarming, and destroying. So the first, you could think that the innate immune system is all the time exerting its role because it prevents the pathogens to get into our body by physical barriers that are considered part of the innate immunity. For example, the epithelium that is present in the respiratory tract, in the urogenital tract, and in the gastrointestinal tract is this lining of epithelial cells that are joined together by tight junctions that impair and provides a physical barriers that impairs the pathogens to get into our body. So that's why if you have a disruption in the epithelium, for example, in the skin, then you can give an opportunity for a pathogen to get in, and maybe you have a little infection and inflammation in the part where you had the skin cut. Okay, so there are various ways to prevent microbes from crossing the epithelium. And so one is the mechanical, and this involves flow in the case of the skin. And again, these barriers that impair the crossing of the epithelium are in the skin, in the gastrointestinal tract, in the respiratory tract, the urogenital tract, and the eyes. And so in all of them, you have these epithelial cells joined by tight junctions that provide a mechanical barrier to impair the entry of the pathogens. But you also have other means that mechanically impair the entry of the pathogen. And that includes, for example, flow of fluid or perspiration. It all in the skin and in the in all of the other tracts and the eyes. You also have in the gastrointestinal tract, respiratory tract, or urogenital tract, the presence of mucus that is secreted by specialized cells, and it will form a layer on top of these epithelium cells that will make it difficult for a microbe even to reach the epithelial cells, okay? So, for example, in our intestine, as we will see, we have tons of bacteria, but not all of them are able to touch the epithelial cells because there is a thick barrier of mucus that is covering the epithelial cells. So this also provides is another example of a mechanical barrier. And then on top of the mechanical barriers, and again, we mentioned epithelial cells, flow of fluid, and mucus, we have chemical barriers. And examples for these chemical barriers are enzymes like lysozyme, for example, that will destroy the microorganisms. And the other chemical barrier is a low pH. For example, in the stomach, in the gastrointestinal tract, there is low pH that will destroy the microorganisms. Again, preventing before it gets into our body, okay? The other chemical barrier is the antimicrobial peptides that are throughout all these surfaces, and is produced in many cases by epithelial cells. And as I mentioned before, these some of these microbial peptides, they can form pores in the surface of the pathogen to promote their lysis. They are also called defensins. And many of them, we are still not sure how they operate. And finally, and so examples again of chemical barriers are enzymes like lysozymes that will destroy the pathogen, are low pH that will also destroy the pathogen, and the antimicrobial peptides. And then we have the microbiological barriers. And again, in this case, is also called normal flora that is all throughout the these surfaces in our body. And this is fascinating, and it has been in the past years an explosion of research into the microbiota, or this normal flora. And it became evident that not all the microbes and bugs are bad. And research from many laboratories have demonstrated that, for example, in the intestine, in the large intestine, the colon, but also in the skin and in other surfaces, we are colonized by large numbers of commensal bacteria. And so it's estimated that the amount of commensal bacteria is exceeds about 10 times the number of cells we have in our body. And so if you think about it, we are more bacteria than what are our own cells in our body. And so there has been experiments done in which it has been shown that the treatment with antibiotics can kill many of these bacteria that coexist with us in our body. And this leads or gives space for pathogenic bacteria to grow and produce toxins that may cause damage to the cells and then cause inflammation in the area where damage exists. And many times these inflammations can become chronic, and this is the case, for example, for colitis, which is a disease that is characterized by dysbiosis, which is an alteration in the composition of the bacteria in the intestinal tract. Okay, and so the conclusion from these studies is that these commensal bacteria that we carry with us normally in a physiological manner, they play a beneficial role for competing with most pathogenic bacteria, and so preventing the outgrowth of these pathogenic bacteria that are able to damage our cells. And in addition to playing a beneficial role, for example, for the processing of nutrients that is necessary for absorption. So, but particularly in terms of host defense and immunity, these commensal bacteria, by competing with pathogenic bacteria for their own nutrients, they prevent the outgrowth of these pathogenic bacteria. There has been also a tremendous amount of research on how these commensal bacteria are able to shape our immune system, and how, for example, bacteria in our gut can modulate constitutive basal levels of immune activation in cells in our spleen, right? That they are far from the gut, T cells from our spleen or macrophages from our spleen, without crossing the epithelial barrier in the gut. And this is because they release small molecules or metabolites that can reach circulation. And indeed, a significant fraction of the metabolites that are not constantly circulating in our body derived from in our blood and reaching as a consequence all the cells in our body derived from commensals that are in our gastrointestinal tract. Okay, I think this is fascinating, and that's why how the bacteria in the gut can have an effect in, for example, macrophages that are far away located from the gut, not only the macrophages that are underneath the epithelium. And again, there has been an explosion of research and understanding on how these commensals alter not only immune-related diseases like colitis, like the response to a viral infection, like the protection against tumor, but even neurological diseases, like there has been some association with autism, for example. And so while more research is needed, it seems that the implications of dysregulation in the commensal bacteria can have consequences even beyond the immune system. Okay, so I would like to introduce to you with this, at this point, some optional learning material. So in April, when all the lockdown had just started, with a colleague in Yale University, Carla Rodling, we initiated a new virtual seminar series in immunology that we named Global Immunotalks. And so these seminars are available free for anyone in the world having internet connection can have access to these seminars, and they are in our YouTube channel named Global Immunotalks. And so particularly today, for those of you that are more interested in this microbiological barrier, the normal flora, and how it can impact the innate immune system, I recommend two seminars, one by Jasmine Belkite at NIH, Microbiota Control of Tissue Immunity, and the other one by Dr. Manuela Rafatellu at UCSD, Nutritional Immunity in the Inflamed Gut. So the links are here, or you can go to the Global Immunotalk, you can check all the videos, and you will find these two listed among them. Okay, any question about that?
Professor, how long are those videos? Like, how long are these talks?
They are about 1 hour, some a few minutes more. What, you know, is up to you. You can watch the whole video and you can watch the introduction. They are very advanced videos. So especially at this early point, don't expect to understand everything about them. Maybe you want to watch part of them, or maybe you want to flag them to watch later in the course, or even later after the course ends. It depends on you. They are definitely optional, and so we will not evaluate any content on these videos. Is for those students that are particularly interested in a topic, and of course we don't have time to cover all the topics related to the immune responses in depth. So you it's an additional optional learning material for those of you that want to put further deeper focus on a particular topic of interest. And I think that the YouTube allows you to speed up, and so it's 1 hour, but you know, you can listen it faster if you wish. It's up to you. But I will be pointing out some of the Global Immunotalks as additional material related to the course topics for those of you who want to watch it. And again, you can watch the first part, which is normally the speakers would give an overview of the topic they will cover. Any question about what I covered in the past few slides, or any other question from the participants? Okay, so let's do another poll here. Let's see if I can, I think I should be able to, okay. So I think that we will do here. So you should be able to see the poll, and this is the question: which of the following pairs is mismatched? And then you have here the options, and I'll give you, let's say 30 seconds to answer the question. And so normally what I advise, you know, it's up to you, right? I'm not there. I don't, you know, you can decide what to do with these questions, but it's a nice way to learn here during the lecture and make the best use of your time if you put effort in these questions that can save perhaps time later studying after the lecture. So I advise that you put effort in trying to answer the questions as well as possible. And it's okay at this point if you need to look at the previous slides. The PDF is in Canvas. It's totally fine. This also provides example, like model of the type of questions that will be in the exam. Okay, so I will end the poll now, and I can share the results while I read the question. Which of the following pair is mismatched? So chemical and antimicrobial peptide, that's correct. That's one of the chemical barrier factors. Mechanical, mucus in intestinal tract, that's correct. Microbiological, the microbiota or normal flora. And so none of the above is mismatched. So you guys, most of you answer the pick the right choice. So we'll stop sharing. I think I can close this, and I will go to the next topic we want to cover, that is the recognition phase of the microbe. And so this recognition phase occurs via soluble proteins, and we will learn the most noticeable and predominant of those ones are the complement proteins and cell-associated receptors. And we will learn that there are membrane-associated receptors and cytosolic receptors. So let's first focus on these soluble proteins. And as I said, the most predominant soluble proteins are the complement system. So this complement system is a system, a group of proteins that are made by the liver and play multiple roles in innate immunity. These complement proteins are present in the serum and the lymph as well, because the lymph is derived from the serum, and extracellular fluid, because the lymph is invading and flowing through extracellular fluid, okay? So from the serum, it can go to the lymph, and so within the lymph, and extracellular fluid. So that means that when a pathogen reaches the blood, the serum complement proteins can recognize the pathogen, but if it doesn't reach the blood, if it reaches only the tissue, for example, the mucosa that is underlying the epithelium in all these different surfaces that we mentioned in previous slides, like the skin of the gastrointestinal tract, then there is complement there as well in the extracellular fluid that can recognize the invading pathogen. And so, again, you should think as the complement as a group of proteins, particular proteins that have a serine protease activity. So they are a group of serine proteases. And so within the complement, we can review the three phases of the innate immune responses, the recognition, the alarming, and the destruction of the pathogen. And so the recognition depends on the pathway of the complement that is activated. The first one to be activated is the alternate pathway, in which the surface of the pathogen will create a local environment that is conducive to the activation of complement, activation of some of these serine proteases, okay? And so there is a lot of chemistry on that I will not go through, and if you are interested, you can read the book, but it will not be evaluated. All what you want to know is that in the surface of the pathogen, there is an environment, chemical environment that is conducive to the activation of some of these complement factors. And so this is the first one to act, because you just need the pathogen surface and the presence of the complement for initiating this activation cascade. The second to act is the lectin pathway, or and the one that can be activated later. And this is mediated by mannose binding lectins. And these lectins will bind to carbohydrates or sugars in the surface of the pathogen and can initiate the activation of complement. And so this is also activated early, and is an additional way in which the complement can be activated. And finally, the classical pathway is the third one to act. And this can be initiated by a C-reactive protein, which is a protein produced by the liver after the liver has been signaled by cytokines produced by the innate response. But most often is activated by antibody complexes. And these antibody complexes will bind the complement, some of these complement proteins, and will initiate the cascade of complement activation. And I'm always talking about antibody complexes here, so it's not just the antibody, but it's the antibody associated with the pathogen or with their ligand in the pathogen, what will trigger the complement activation. So it's the antibody complex, antibody plus pathogen, what triggers the complement activation. And so I have a question for you now. This will be the last one to be activated. This will be a slow way to activate the complement. Why do you guys think this is the slowest one? Any brave one?
Is it because, I have a potential, so is it because it requires the adaptive immunity?
Yes, excellent, excellent. That's the way I want you to study, by thinking, so this, we mentioned that it requires antibody or C-reactive proteins that are produced by the liver later in the immune response. But most often is antibodies, and antibodies are derived from the adaptive immune response, and we mentioned how the adaptive immune response is slow. So these two are the first ones to be activated early on, but all of them can lead to complement activation. And this complement activation will always converge in one central, a common event, that is the cleavage of this factor that is central. It's called C3, complement factor 3. And it will be cleaved to produce two products or two pieces that are called C3A and C3B. And so these products will lead to the recruitment of inflammatory cells, and so alarming the rest of the body that there is a presence of the pathogen, or at least neighboring cells that there is a presence of the pathogen. And so this is induced by C3A together with C5A, and as we will learn, and will also result in the opsonization of pathogens, facilitating uptake and killing by phagocytes like macrophages. And this is in this opsonization, or this enhancement of pathogen uptake, is mediated by C3b, and it will also induce a pore formation in the surface of the pathogen that will lead to the destruction of the pathogen, and this is initiated by C5b, okay? So we will go in detail now through each of these outcomes, but I want you to have here with this slide an overview of complement activation. So we said that it can be initiated by the recognition of the pathogens through alternative pathway, lectin pathway, or classical pathway, and in all the cases will converge into cleavage of C3 to C3a and C3b, and this will then result in alarm of the body that there is a pathogen by recruitment or of inflammatory cells, or by destruction of the pathogen by opsonization, uptake of the pathogen, or by perforation of pathogen membrane. Okay, so here
Professor?
Yes.
Sorry. So are complement proteins just any proteins that form the complex that results in performing that immune response of the alarm, destroy?
No, they are not any protein. They are a group of proteins that are for serine proteases that form part of this complement system.
Okay, thank you.
Okay, so here we have this central event that we mentioned in the previous slide, the cleavage of C3, the complement factor 3, into C3a and C3b, okay? Whatever way the complement is activated, it will always converge into this cleavage of C3, okay? And so we learn how the C3a is important for recruitment of pathogen, and I have a slide showing that. But the C3b, which will now covalently bind to the pathogen surface and tag the pathogen for destruction, okay? And we will learn more about that. Once the C3b binds to the pathogen, it will also initiate another important event in the complement activation that I want you to remember, that is the cleavage of C5 into C5a and b, okay? So this is what here I want you to remember, that once C3b is produced, it will bind to the surface of the pathogen. In the meantime, C3a will be released and will act recruiting inflammatory cells like phagocytes, cells like macrophages and neutrophils, and we will see that in the next slide. But importantly, this C3b, now bound covalently to the surface of the pathogen, will induce the cleavage of C5 into C5a and C5b, okay? Okay, keep that here in mind. So we talked that C3b will induce the destruction of the pathogen. One of the ways in which it will induce the destruction of the pathogen will be by enhancing or increasing the pathogen uptake. And this is called opsonization. So an opsonin is any molecule that enhances phagocytosis by marking an antigen or a foreign presence for an immune response, okay? This is an opsonin that will mark any a factor or pathogen for an immune response, and this is called an opsonin. So the C3b will mark these bacteria for enhancing their uptake by phagocytic cells, and so it's an opsonin that will be inducing this opsonization, that is the increase of pathogen uptake. So here you have a cartoon showing that the complement activation leads to the position of C3b on the bacterial cell surface. And then these macrophages that we mentioned in previous slides that are specialized in phagocytosis, they have a receptor for the complement C3b. And so they now can be better at uptaking these bacteria because they can see the bacteria better by their complement receptor that binds to C3b and will induce the endocytosis of the bacteria by this macrophage. And then this macrophage membrane will fuse, creating a membrane-bound vesicle that is called the phagosome, that then will bind to the lysosomes that have much lower pH and forming the phagolysosome, where there are enzymes and reactive oxygen like lysosine and reactive oxygen and nitrogen species that will destroy this pathogen, okay? And so this is how the C3b, by inducing opsonization, which is an enhancement in the uptake of the pathogen, will lead to the destruction of the pathogen. So we also talk about C3a promoting the recruitment of immune cells. And it's not only C3a, but also C5a that we mentioned in previous slide is also produced upon complement activation. They will both induce the recruitment of immune cells. And one way they do it is because they are able to increase the vascular permeability. And what is that? So in our vessels, we have a specialized epithelium that is called endothelium, endothelial cells that are also joined together by tight junctions. And so the C3a and the C5a will increase the blood, dilate these vessels by inducing the weakening of these tight junctions. And so the vessels will become bigger. And this increased permeability now, so the content of the vessels will be able to permeabilize out from the vessels. And this increased permeability of the endothelium will allow increased fluid leakage from blood vessels and extravasation of complement proteins and other plasma proteins to the side of infection where complement has been activated and where these factors have been released, okay? So not only that, but in addition, these factors are chemotactic. So they will attract migration of monocytes and neutrophils from the blood into the tissue. And these, as we mentioned, will uptake pathogens via phagocytosis, and they will destroy the pathogens. And so these cause, all initiated by this increase in blood flow due to weakening of the tight junctions in the endothelial cells, cause an inflammation. And this is very important, and I want you to remember this term. Inflammation is characterized by heat, redness, and swelling and pain. And so you should remember the three features of inflammation. And not only remember the three features of inflammation, but understand very well why they, how they are caused. And so when you have an increase in the blood flow, and in this case, this is induced by C3a and C5a, but we will learn that there are many factors that increase this vascular permeability, so they weaken the tight junctions in the endothelial cells to increase the blood flow. This increased blood flow in these vessels will cause heat and redness, okay? So that makes sense because there will be more blood in these vessels that are vascularizing the tissue that is where the pathogens are, right? Or where there is being triggered an inflammation because initiated by the increased vascular permeability. And so in this area of the tissue, and you may have experienced that, there are increased heat and redness that is caused by the increased blood flow, okay? And in addition to that, we have learned that there will be extravasation of plasma proteins and also cells into the tissue. And what will that cause? If you have more proteins and more cells that go out to the in the site of inflammation into the tissue, what would be the consequence of that?
Swelling.
Swelling. And so you should be thinking that makes sense that you will have swelling in the inflamed tissue. And this is because you have more plasma proteins and plasma and blood cells that are getting into the tissue. And that crowdiness in the tissue will make pressure into nerve endings that are in the tissue. And that causes pain, okay? So what you want to remember is not only the 4 features of inflammation, but you need to make sure you understand how they are caused. And so again, heat, redness, swelling, and pain, which in Latin are calor, rubor, tumor, and dolor. Okay, you don't need to remember the Latin.
Professor, I don't know if this is an incorrect way of thinking about it, but I was wondering if the permeability increase is just caused by that dilation. Like, do we need to know, like, is there anything mechanically happening that's different from this dilation of blood vessels to dilation caused by like by something else, or C3A and C5A dilation of blood vessels allowing this permeability and causing the heat, redness, swelling, and pain to get rid of the infection or whatever it is? Is that somehow mechanically different than, let's say, our heart, the parasympathetic nervous system and dilating our blood vessels? Like, is there a difference mechanically?
Yeah. I mean, I think that there are differences in which it is induced, but always the dilation of blood vessels would cause increased permeability. And so we will learn not only C3A and C5A are causing this, but there are other factors like TNF-alpha, for example, that are cytokines that can also cause increased permeability and result in inflammation.
Thank you. All right.
Okay. So this is how the C3A and also the C5A will alarm because they will allow increased permeability for plasma proteins and cells to come, and they are also chemotactic, right? So they will attract immune cells into the site of inflammation. So that may be a difference, for example, from a dilation that is induced just by the autonomous nervous system, okay? So the other mechanism we mentioned, the complement will lead to destruction of the pathogen, is via the lysis of the pathogen. So in this case, the factor that will initiate that is the C5b that we mentioned that will be produced in the complement activation. And this leads to a protease cascade. And again, I don't need you to remember all these factors, but ultimately to a formation of a pore in the surface of the pathogen that will allow the entry of nucleases and proteases into the pathogen and induce the lysis or death of the pathogen. And so again, the core slide here, the complement can be activated by the alternative pathway, the lectin pathway, and the classical pathways that by recognition of the pathogen in all cases will converge into 3CA and into C3 cleavage that leads to the production of C3A and C3b. Remember, C3b binds covalently to the surface of the pathogen, and that initiates the cleavage of C5 into C5a and C5b, okay? And then these products that I just mentioned will have consequences that will lead to recruitment of inflammatory cells, opsonization, and perforation of the pathogen cell membranes that will all lead to the elimination of the pathogen. Okay, let's, yes, please, yeah, Lucas, go ahead.
So I was wondering, regarding the classical pathway, does that mean you're only going to be able to have, because if you're responding to a disease for the first time, that means the only way you're going to be able to fight it is through the classical pathway, because you have to wait for the adaptive immune system to create. No?
Good question, but no. The complement doesn't need to have seen the pathogen before. The
so just act.
innate immune system doesn't need to, it's ready to act. You know, actually it's acting in your body right now by preventing pathogen entry, but it's ready to act. As soon as you have a cut in your skin, the pathogen will have now a chance to get into, microbe will have a chance to get into the your tissue, and the complement is there, it's in the extracellular fluid. And so will start being activated just by binding to the pathogen surface. That will be the first one that quickly will trigger in minutes a cascade of complement activation. Okay.
Right. So there doesn't need to be some sort of like previous recognition. It just needs to literally be any contact. Okay.
Any contact is, and that's in general how the innate immune system works. You don't need to have pre-exposure. Actually, for the adaptive immune system, you don't need pre-exposure either.
Oh, yeah.
But it's true that the adaptive immune system generates memory response, which the memory response, the secondary responses require pre-exposure, and they are better at eliminating pathogen. Okay, but your immune system is ready to. The first time they see a pathogen, the immune system can respond. Okay. Yeah, sure. Okay, let's do a poll here. Okay, so let's stop the poll and share the results. And so which of the following is not a characteristic of inflammation? Okay, so redness and swelling are, and production of functional BCR or B cell receptor, we will learn later it has nothing to do with inflammation, and actually it occurs even before the infection, so the B cells can be there ready to recognize a pathogen via the BCR, okay? So I stopped sharing. So So I tried to find a watch, but I couldn't. So how much time do I have? Anyone? Okay.
Technically.
Let's see. If not, I can check, but I didn't want to go get out. Okay. Okay, perfect. Thank you. Okay. Okay, so 5 minutes left. Okay. Okay, so let's, I covered much less. I think I got too excited with some parts, but I covered much less than what I thought. But that brings me the opportunity to tell you that what we will cover in these lectures and the schedule in the lectures is tentative. And so we go at any pace that we think is necessary. And so you guys don't worry too much about the lecture schedule. I may update it in the syllabus as before the exam based on what we cover, but you focus on the slides that we will be covering during the lecture. Okay, so so we learned then how the complement or soluble proteins, extracellular proteins are important for recognizing the microbe. But the microbes can also be recognized by cell-associated receptors. And what you want to remember is that these cell-associated receptors, they are always, they are called also pathogen recognition receptors, or PRR, and they will be binding patterns or patterns in the pathogens that are present in a group of pathogens, and they can be distinguishable from the self proteins or factors. And so these ligands in the of the pathogen recognition receptors are called PAMPs, or pathogen associated molecular patterns, and I will learn more about them. So always a PAMP, which is in the pathogen, and it's basically a feature in the pathogen, a pattern in the pathogen, will bind to these pathogen recognition receptors, PRRs, and then via an adaptor protein will induce intracellular signaling that will lead ultimately to production of factors that will either alarm or destroy. Okay, so this is a very general view, but you will see how it's helpful that you remember that it's always a pump in the pathogen binding to a pathogen recognition receptors via an adapter, inducing signaling that leads to production of factors that alarm or destroy. And so let's go, let's see. I think that what we will do is we will go directly to the summary slide, and we will summarize a little bit what we were able to cover today. And next lecture, and probably next 2 lectures, we will finalize covering the innate immune system. And so let me find the summary slide here, if I may, here, okay. So, so we were able to cover the generalities of the innate immune response, which is rapid, is present in animals and plants. They have limited specificity, so they recognize PAMPs. Now we introduce a terminology, these pathogen-associated molecular patterns that are present in a group of pathogens, right? And they are encoded in germline, the receptors that recognize the pathogen patterns, okay? These receptors of the innate immune system. We learn about the innate immune responses being able to occur to some level in all cells of our body. but we also learn about some professional innate cells. And we mentioned that we will learn more about basophils, eosinophils, and mast cells that are important for parasite defense and play a role in allergies later in the course. And today we cover mostly the role of neutrophils that phagocytize and do netosis, macrophages that phagocytize and can destroy the pathogens and be empowered by natural killer cells and T cells, dendritic cells that not only release factors that limit the pathogen, but are important antigen-presenting cells, are the most powerful antigen-presenting cells. And natural killer cells that we will learn more in the next 2 lectures. And then we learned that the innate immune system acts by preventing infection we talk about physical barriers, mechanical barriers, or physical mechanical barriers, chemical barriers, and microbiological barriers, and we talk about the normal flora. And we also mentioned that once the infection breached these barriers and it could not be prevented, then the innate immune system has ways to quickly recognize the presence of the pathogen with soluble proteins like the complements, and we learn a lot about the complement today, how it can be activated, which is the central core event, the C3 cleavage, and which are the outcomes of this event. And we just mentioned that the other way in which the recognition can occur is through cell-associated receptors that are called PRR, pathogen recognition receptors, that will always bind a PAMP, a pathogen-associated molecular pattern, to induce via an adapter a signaling that results in the production of factors that will then alarm and destroy, okay? So next lecture, we will continue with what was not covered today and furthermore, and I foresee that there will be 2 more lectures about innate immunity.